Have you ever wondered why the size and shape of your medicine matters in its effectiveness? Welcome to the fascinating world of Micromeritics, a scientific study that dissects the physical properties of minuscule particles and holds a pivotal role in the pharmaceutical industry. So, what makes Micromeritics so important, you ask? Well, imagine this. You're taking a medicine that's supposed to dissolve in your stomach and enter your bloodstream. If the particles of the medicine are too large, they might not dissolve as efficiently, leading to a delayed or reduced effect. On the other hand, if they're too small, they might dissolve too quickly, causing an immediate, possibly overwhelming effect. That's where Micromeritics comes in. It helps us understand and control the size and shape of these particles to ensure the medicine works as intended. But it's not just about size and shape. Micromeritics also dives into other properties such as particle volume and number, and surface area. For instance, knowing the particle volume can help us predict how the medicine will behave in different conditions. Similarly, understanding the surface area can give insights into how quickly the medicine will be absorbed in the body. And it doesn't stop there. Micromeritics also provides us with reliable methods to measure and determine these properties. These methods are vital in ensuring the consistency and effectiveness of drugs, making Micromeritics an indispensable tool in drug development and quality control. So, as you can see, Micromeritics plays a crucial role in the pharmaceutical world. It's the unsung hero that ensures your medicine does exactly what it's supposed to do, when it's supposed to do it. It's the science behind the pill that helps you sleep, the syrup that soothes your cough, and the injection that saves lives. In the world of pharmaceuticals, Micromeritics is the key to unlocking safer, more efficient, and more effective medicines. Now let's delve deeper into the significance of the particle size and its distribution. Imagine you're dissolving sugar in your morning cup of tea. The smaller the sugar granules, the quicker they dissolve, right? This is because smaller particles have a larger surface area relative to their volume, which allows for a faster dissolution rate. Now think about applying this principle to medications. The size of particles in a drug can significantly influence its dissolution, and consequently, its absorption into the bloodstream. Smaller particles typically dissolve more rapidly, resulting in quicker absorption and onset of therapeutic effects. However, it's not just about size alone. The distribution of particles is also key. Imagine you've got a bag of mixed nuts. If you blindly reach into the bag, you want to be sure you're not just getting a handful of peanuts every time. Similarly, for a drug to be effective, it's important that the active ingredients are evenly distributed. This ensures that each dose will have a consistent concentration of the medication. If the particles are not evenly distributed, some doses might be more potent than others, leading to inconsistent and potentially unsafe effects. So in the realm of pharmaceuticals, achieving the right particle size and distribution is a fine balancing act. It involves careful formulation and processing to ensure that the drug not only works effectively, but also provides consistent dosing. This is particularly crucial for medications that are potent or have a narrow therapeutic window, where small variations in dose can have significant impacts on safety and effectiveness. In the pharmaceutical industry, numerous techniques are employed to achieve the desired particle size and distribution, including milling, micronization, and spray drying. Each of these methods has its own strengths and limitations, and the choice of method depends on factors such as the nature of the drug, the desired dosage form, and the intended route of administration. The size and distribution of particles can significantly impact the effectiveness of a drug. So, the next time you take a medication, remember that there's a lot more to it than just swallowing a pill. It's a science, and a pretty fascinating one at that. Did you know the shape and volume of a particle can also influence a drug's effectiveness? Quite intriguing, isn't it? Let's delve into this a bit more. Particle shape is a crucial parameter in the pharmaceutical industry. It influences flow properties, which in turn affect the manufacturing process. A more rounded particle, for instance, will flow better than a more angular one. This smooth flow is essential in processes like tablet compression and capsule filling. Now let's talk about volume. Particle volume is directly proportional to the drug dosage. This means that larger particles will deliver a higher dosage of the drug, while smaller particles will deliver a lower dosage. Managing particle volume is a fine balancing act to ensure the right dosage. In essence, the shape and volume of particles play a significant role in determining the final product's quality, effectiveness, and safety. Indeed, the shape and volume of particles are important factors in drug development. What about the surface area and the number of particles? How do they influence a drug's properties? Well, let's dive in. The surface area of a particle is incredibly significant in the world of pharmaceuticals. When a drug enters your body, it needs to be absorbed to exert its effect. This absorption usually occurs across the surface of the drug particles. The larger the surface area, the more space there is for the drug to be absorbed. Think of it as a sponge. The more surface area it has, the more water it can soak up. Similarly, the greater the surface area of a drug particle, the more readily it can be absorbed into your body. Now you might be wondering, how can we increase the surface area? Well, one way is by reducing the size of the particles. Smaller particles mean a larger surface area relative to volume, leading to quicker and more efficient absorption. And it's not just about absorption. A larger surface area can also enhance the drug's dissolution rate, which is critical for the drug's bioavailability. Let's shift gears and talk about particle number. In the realm of pharmaceuticals, it's not just about how big or small the particles are or how much surface area they have. It's also about how many particles there are. The number of particles can determine the dosage of a drug. For example, let's say we have two drugs with the same total mass, but one has larger particles than the other. The drug with the larger particles will have fewer particles in total. This can affect the distribution of the drug within the body and potentially its effectiveness. Hence, controlling the number of particles is another key aspect in the formulation and dosage of a drug. So we can see that both the surface area and the number of particles play a crucial role in the effectiveness of a drug. They influence how quickly and efficiently a drug is absorbed, how it is distributed within the body, and how much of it is required to achieve the desired effect. So the surface area and the number of particles are also key factors in the effectiveness of a drug. Now, you might be wondering, how do scientists determine these particle properties? Well, let's dive into some of the methods employed to measure particle size and volume. Firstly, we have the sieving method. Picture a stack of sieves with different mesh sizes. The largest mesh is on top and the smallest at the bottom. The sample is placed on the top sieve and the stack is mechanically agitated. Particles larger than the mesh size of each sieve are retained, and thus, by weighing the amount in each sieve, we can determine the particle size distribution. Simple, yet effective. Next, we have microscopy. In this method, a microscope is used to directly observe and measure particle size. This can be done using either an optical microscope for larger particles or an electron microscope for those really tiny particles. Now, while this method can be time-consuming and requires significant skill, it provides an unparalleled level of detail. Then there's the sedimentation method, which is based on Stokes' law. Here, the rate at which particles settle in a liquid is measured, and this information is used to calculate particle size. However, this method is best suited for particles that are spherical and have a narrow size distribution. Lastly, let's talk about laser diffraction, a method that has become increasingly popular in recent years. This technique involves shining a laser through a sample and then measuring the angle and intensity of the light scattered by the particles. The data is then processed to calculate particle size distribution. The beauty of this method is that it can handle a wide range of particle sizes and provides rapid results. Particle volume, on the other hand, can be determined using methods such as cycnometry, gas adsorption, or mercury intrusion porosimetry, each with its own set of advantages and limitations. So, there you have it. From sieving to laser diffraction, these are some of the methods used to measure particle size and volume. These methods provide crucial data that scientists use to develop effective drugs. So, as we've learned, micromeritics is a fundamental science in the pharmaceutical world. It gives us the tools we need to understand the behavior of particles, from their size and distribution to their shape and volume. We've delved into the importance of surface area and particle number, and how they affect drug delivery and efficacy. We've also explored various methods for determining particle size and volume, giving us the ability to control and optimize the characteristics of our pharmaceutical products. These are critical factors in the development of effective, safe, and reliable medicines. In conclusion, The study of micromeritics is crucial in the pharmaceutical industry. It provides a foundation for understanding and manipulating particle behavior, leading to significant advancements in drug development and treatment strategies. So don't stop here. Dive deeper into the intricacies of this fascinating field. Subscribe for more because you won't find me later. Stay curious and keep exploring the fascinating world of pharmaceuticals.